Hello from somewhere on the beautiful Puget Sound in the Pacific Northwest. My name is Giancarlo Bonora. I'm going to present the results of a study conducted on thousands of individual cells looking at chromatin conformation, accessibility, and expression during early mouse development. This is work that I conducted as a postdoctoral fellow in the Noble Lab in the Genome Sciences Department at the University of Washington in Seattle, in collaboration with the Distish and Deng labs and many other UW colleagues. A manuscript based on the results of the study is available on bioRxiv and has been accepted by Genome Biology for publication. So we all started out as a single cell or zygote, which is totipotent. And even after several rounds of cell division, the cells of the early mouse embryo or early mammalian embryo, known as embryonic stem cells or ESCs, largely retain this capacity, this pluripotent capacity to become any cell type in the body. And then during the course of development, 3 tissue layers emerge: ectoderm, mesoderm, endoderm, representing more differentiated states. And the ESC or embryonic stem cell nucleus is known to undergo dramatic reorganization during the course of early development, as exemplified by, in this micrograph, by during the differentiation to neural precursor cells, one sees a dramatic accumulation of compact heterochromatin as visualized by these yellow regions in this micrograph. Another example of extreme genome reorganization during development is what occurs to the inactive X chromosome in female cells. So initially in embryonic stem cells, both X chromosomes are open and active in female mammalian cells. And then during the course of differentiation, one X chromosome at random becomes inactivated and assumes a very compact structure. HitSeq is a DNA sequencing technique that captures 3D interactions or pairwise contacts. And this is often visualized as a heat map or contact map representing all the frequency of interactions for all pairwise regions along the genome. And here I'm showing an example for a single chromosome. And what each row and column represents is all the interactions made by a single locus with all other loci in the genome. And here I'm showing an example of a contact map for the active X chromosome. And you can see structural features that are typical for autosomes. In contrast, the structure of the inactive X has a bipartite nature with 2 superdomains representing regions of preferential interaction. So it has a very distinct structure relative to the active X autosomes. For the study, we had 3 main biological questions. Firstly, we wanted to find out how the inactive X bipartite structure is established within individual cells during the course of the cell cycle. We had the same question during the course of differentiation and cell division. And thirdly, we wanted to relate gene silencing, so expression and accessibility, to this inactive X bipartite structure. For the study, F1 hybrid mouse embryonic stem cells represented the starting state of the study, and this allowed us to get, the F1 hybrid system allowed us to get allelic information. And cells were differentiated towards embryoid bodies and sampled at 4 time points. Day 0 representing the embryonic stem cells, day 3, day 7, and day 11. And then from the day 11 cells, MPCs were clonally derived, and these represented the fully differentiated state where it is known that one X chromosome had been activated. And then from the samples, we conducted single cell HitSeq, single cell RNA-seq, and single cell attack sequencing. It is known that compartmental structure in the nucleus emerges during interphase, as exemplified by the 2 chromosome 1 homologs in the lower panel of this figure. And one sees something similar for the active X chromosome as shown in the top right panel. In contrast, we found that for the inactive X chromosome, one sees the reemergence of the bipartite structure during the course of the cell cycle. Using single cell or allelic single cell HitSeq data from the time course experiment, I performed a trajectory analysis and obtained this branch structure shown on the slide. Here, every data point represents a single cell colored by time point, and what we observed is that there's an early branch, an intermediate branch, and differentiated branch. So this trajectory appears to capture the 3D conformational state of different cell states during development. Using the single cell IC data, we also found that the proportion of cells with the inactive X chromosome increases during the course of development. Similarly, the proportion of cells with a silenced X chromosome increases during the course of development, both by allelic expression, single cell expression analysis, and by allelic single cell accessibility analysis. And taking this data together, we found that the proportion of cells with a bipartite X tracked the proportion of cells with a inactivated X chromosome or silenced X chromosome, but that the changes in the structure appear to lag the silencing of the X somewhat. Thank you very much for listening. Please see my poster for further details. I've only given a very small vignette of what we show in the paper. Thank you very much.